plant frequency using nested plots and analyzing frequency data. Why does plot size matter when we're concerned with taking frequency estimates? This is because frequency estimates are directly related to the size of the quadrat or plot that is used for measurements. Frequency is defined as the number of times a plant species is present in a given number of uniformly sized quadrats placed repeatedly across a stand of vegetation. The definition specifically refers to quadrat size because whether or not a plant is present or absent in a frame or quadrat clearly depends on the size of the quadrat. Please note that throughout this presentation, I will be using the terms quadrat, plot, and frame interchangeably. All of these terms refer to the area in which our estimates are taken. We can further examine the question of why plot size matters by looking at a schematic diagram of vegetation and looking at how our estimate of frequency for the pink daisy changes as we change quadrat size. It's clear that the frequency estimate for pink daisy increases as the size of the quadrat increases. And this increase is observed because larger plots cover larger areas. So we have a higher likelihood of capturing or encompassing more plants in larger size quadrats. Now you may be wondering, that's nice, why should I care? And we need to remember that we are using frequency data to detect change in vegetation over time. But frequency data are different than other types of data that are associated with plant attributes, such as density, cover, or biomass. Frequency data are discrete because we are focusing on the presence or absence of plants in the plots. And so that they are binomial data and follows a binomial distribution. With binomial distributions, our ability to detect change is greatest in the range between 40 to 60% frequency. It's also still very good in the range between 20 to 80% frequency. But in the lower reaches, or the more extreme reaches, such as below 10% or above 90%, it's not as sensitive to change. When we're talking about change, we're talking about changes over time. So if we're comparing a frequency estimate taken at year 1 and a frequency estimate taken at year 2, we cannot detect change in these lower regions. Now, we know that vegetation change happens, and that's why we're measuring frequency in the first place. So frequency is going to change. We need to avoid using quadrat sizes that produce frequency estimates in these extreme ranges. Now first, let's talk about how we determine the correct quadrat size in the first place. The way we do this is by repeatedly estimating frequency using increasingly larger and larger quadrats. And we estimate frequencies for those species that we're interested in. Now we don't actually go out and repeatedly place different size quadrats on the ground. Generally what we do is we use nested quadrats. And what this means, a nested quadrat, is a frame that has marks, or sometimes it may have some hard edges inside it that delineate different size quadrats inside a single frame. So here we see a nested quadrat that has 4 different quadrat sizes nested within the largest quadrat. And we can look at the difference in frequency estimates that we get for different species. The grass species is the most common species in this area that we're looking at, the vegetation that we're looking at, and we can see that in the smallest size quadrat, that's this small size here, grass occurs in 2 of the 5 quadrats. And so we detect a 40% frequency using the smallest quadrat. By the time we get to the next larger size quadrat, quadrat size 2, the frequency has skyrocketed to 100%, and it remains at 100%. For the yellow flower, we do not detect any yellow flowers in the smallest size quadrat, but we do begin to detect yellow flower in quadrat size 2, and the frequency of the yellow flower increases as the quadrat size increases. For both the white flower and the clover, we don't detect it at all in the 2 smallest size quadrats, but we do, when we do start to detect it, is in quadrat size 3, and again, the frequency for these species is going to increase with larger size quadrats. In order to determine the correct quadrat or plot size, we need to select the appropriate plot size for each of the individual species. When we are comparing frequency or looking for changes from year to year, we always do this on a species by species basis, and we don't compare frequency between species. We need to maintain a consistent plot size over time, and we compare changes for species only if the same plot size is used consistently. So what do we do if the vegetation changes and the quadrat size is no longer ideal or appropriate? This is where we run into trouble. We know that vegetation is going to change, and that's why we measure frequency in the first place. The first thing that we would do is we would adjust the quadrat size as needed. We measure or estimate the frequency of the species in the original quadrat size as well as in the new quadrat size. By doing this, it allows for continuity and comparability between years when we start to use different size quadrats. One of the problems that we have is that this approach takes time to measure species frequency in multiple frames, and it also assumes that we know that we're near a size threshold. Yet often we don't know this until we've already gone out into the field and collected data and then started to analyze the data back in our office. So here's how we can use nested quadrats to deal with this issue. First of all, we start off with a frame that has some type of a barrier or marking on it, and we have different plot sizes within this frame. So we delineate different plot sizes. We have the smallest plot size 1, which is contained in plot 2, the next largest size plot. Plot size 3 is larger still and contains plot 1 and plot 2. And then the whole frame, the largest plot size, is considered plot 4, and it includes all of the other quadrats. That's why we call these nested quadrats. We use the nested quadrat to record the frequency in the smallest plot size. So let's say, for example, we were looking at determining the frequency of clover, and we see that it occurs in plot 2. It also occurs in plot 3 and in plot 4. So we would record this as occurrence in plot 1, and by extension, we know that it occurs in plot 3 and plot 4, but we would only record the smallest number. This is a dynamic approach, and it facilitates comparison even if plot sizes needs to change. So by using nested plots for measuring frequency, we can then compare year after year. So let's look at an example to see how this would work. Here we have an example in which we have placed 4 nested frames, sample A, sample B, sample C, and sample D, in our environment. We're measuring simply 2 species, and we see that for the clover, in sample A, the smallest plot it occurs in is plot size 4. The grass, on the other hand, occurs in plot size 1. In sample B, the clover occurs in plot size 3, and the grass occurs in plot size 2, and on. For sample C, we see that the smallest plot in which clover occurs is plot size 3, and the grass occurs in plot size 1. And finally, in sample D, the smallest plot that clover occurs in is plot size 2, and the smallest plot that the grass is observed in is plot size 3. To look at how this is analyzed then, we look at the number of hits and calculate the frequency in each of the different plot sizes. And remember, our goal here is to determine the ideal plot size for measuring each of the individual species. So for the clover, we see that we have, with plot size 2 and 3, we have a frequency between 25% for plot size 2 or 75% for plot size 3. For the grass, we're able to detect a frequency of 50% with plot size 1 and 75% for plot size 2. In an ideal world, because we have this overlap, we could use plot size 2 to calculate or to measure frequency for both of these species. Now, in the real world, we would not make these assumptions using just 4 samples, but this shows how you would determine the appropriate plot size for each of the species. Now let's switch gears and examine how we analyze and interpret frequency data. Remember, frequency data are used to assess vegetative change, either at one site over time, so between measurement periods, or to compare vegetation that is between sites. Frequency comparisons are always done on a species-by-species basis, meaning that we compare changes within a single species and not between 2 species. The 2 most common statistical approaches are to use binomial confidence intervals or the chi-square analysis. We would use binomial confidence intervals because data are discrete and they have a binomial distribution. And a chi-square analysis is well suited to binomial data. Binomial confidence intervals are based on a desired confidence level that is stated, our sample size, and the observed frequency. They're calculated as the square root of p times q divided by n. In this case, p represents the proportion of plots that contain the species of interest. Q is the proportion of plots without the species, so that equals 1 minus p, and n is the number of plots that observed. We then take the square root of this value, p times q divided by n. Now, it is important to note that the use of binomial confidence intervals is based on the assumption that each of the quadrats is selected at random and is independent of the others. If you may recall from the previous presentation, these quadrats that we use for frequency are usually located in the environment systematically, and so they're not located at random. However, if we space the quadrats far enough apart that we can ensure that they're going to be measuring different plants and different plant patches, that helps us to meet the assumption of independence. So let's take a look at an example where we use binomial confidence intervals. In this example, we've measured blue grama frequency in 2 different years using 200 quadrats. And in the first year, we estimated the frequency at 34%, or written as proportion, 0.34. In year 2, the frequency of blue grama was recorded as 40%, or written as a proportion, 0.40. And using this formula, the confidence intervals that we would obtain for each of these years are between 27 to 41% for year 1, and between 33 to 47% for year 2. Confidence interval tables can be obtained from Despain et al. And in other resources as well. Now, in order to determine whether we've got a real change, the question would be, without the confidence interval, you might say, well, is 34% different than 40%? And by looking at these 2 confidence intervals, the first one for year 1 and this one for year 2, we would say that there is no difference in blue grama frequency because the confidence intervals overlap. So we don't have to worry about whether 34% is different from 40%, but the fact that the 2 confidence intervals at a 95% confidence level, the fact that they overlap, we would say with 95% confidence that there has not been a change in blue grama frequency from year 1 to year 2. Now let's look at using at a chi-square analysis, and we will look at the same example with the chi-squared analysis. With a chi-square analysis, we want to compare observed values, those values that we obtained in the field, to expected values. We're testing a null hypothesis that there is no difference between the observed frequencies that were detected in 2 different years. and we use a 2x2 contingency table for chi-square analysis. Here we see an example of a typical 2x2 contingency table for present and absence, and we have our observed proportion here for both presence and absence, and also the expected proportion for presence and absence. Now, I'm not going to go into the details of the calculations in this presentation because it is covered very thoroughly in the learning guide. But we use the chi-squared formula given below and calculate a chi-squared statistic. We then compare that to a critical chi-squared value from a chi-squared table. So let's look at an example of how this is done using the same example that we had before of Blue Grama frequency estimates. Our observed proportion, 68, we had 34% frequency out of 200 quadrats. So we had 68 was the proportion and 132. That gives us, that was in year 1. And then in year 2, we had 80 where they were observed out of 200 and 120 that were observed out of 200. The expected values are 74 and 126. If we plug these data into the chi-square formula, we have 68 is the observed present minus 74, which is the expected, divided that by the expected. This value is squared first and then divided by the expected. That's for Blue Grama presence. We do that for Blue Grama observed presence in year 2. We also do it for the absence data in year 1 and 2, and that gives us a chi-squared statistic of 1.56. We compare that chi-square statistic, 1.56, to a critical chi-square value from the chi-square table. The critical value of 2.706 is greater than 1.56, and therefore we do not reject the null hypothesis. The frequency of Blue Grama, based on this chi-square analysis, did not change between year 1 and year 2. As you may recall from the binomial confidence intervals, we reached the same conclusion. So, to conclude, frequency estimations are greatly affected by plot size. The sensitivity to change is best when the frequency estimates are between 20 to 80%. We need to use the appropriate plot size to keep frequency estimates in this range, and we use nested plots if needed. In order to compare our estimates of frequency to detect change, we can use either a chi-squared analysis or binomial confidence intervals.